From when I came here until we thought about setting up the Department of Human Genetics, we had several very good human genetic faculty members that were scattered among different departments. So from the administration point of view, it looked like we were pretty well covered in human genetics. But there were emerging areas that we were very weak in, like statistical genetics. This was prior to GWAS studies and things like that, but that was coming, and the key about doing those population-based studies is you need a lot of statistical geneticists. And there was no department that felt like it was their responsibility to cover all the bases for human genetics. And so it seemed like that was something that was needed at Emory to really propel the field here. So in 2001, we started the Department of Human Genetics. And then very soon after that, we then took over all the clinical activities in human genetics as well by moving the division of medical genetics from the Department of Pediatrics into the Department of Human Genetics. And the Emory Genetics Laboratory, which is the diagnostic lab that was in the division, moved with the division into this department. Diagnostic genetics at the time was undergoing a transition that had been heavily cytogenetic in focus, heavily prenatal in focus, towards a more molecular testing as more and more genes were identified that cause human disease. So it seemed like the Emory Genetics Laboratory could expand itself to fulfill that need of diagnostic testing, particularly in the molecular arena, that would both help patients and the clinicians. And that's what we did. When we took over EGL, it was small but well run. It was, like I said, undergoing this transition and focus. But it was predominantly present to serve the needs of the local geneticists. But we soon saw that there was a need nationally and now internationally for a laboratory like EGL to offer diagnostic testing. And so as the molecular revolution expanded in human genetics, there was more and more testing to do, more tests put online as diagnostic tests, that it always seemed like there was more room to grow. Back in the early days, it was single gene tests predominantly, and we were looking just to add more single gene tests. Then we went to, more recently then, to the idea of panels, where taking advantage of the knowledge that there may be several genes that ultimately can lead to a very similar phenotype that allows the clinician to be able to diagnose the phenotype clinically, but not have to pinpoint what gene it was, but rather order a panel that would cover those genes that have been found responsible. And now it's evolving again into whole exome sequencing, or medical exome sequencing, that covers all the genes that are known to be responsible for genetic disease. And ultimately, it will probably go to whole genome sequencing as well. I think that introducing next generation sequencing to the clinical arena provided a fuller coverage of genes that can be examined as being responsible for a particular phenotype. And I think that was pretty well appreciated by the clinicians. Emory does a good job of freeing the data, or putting our diagnostic results out and available for anybody to see, because that helps all clinical laboratories in the world to interpret their own results. And in the past, if laboratories keep that information to themselves, they very, very slowly build up the database, but this way it can explode very quickly. And we can just, again, provide better service for the clinicians and better service for the patients. I first realized that EGL had a potential to be a world-class laboratory when we started attracting laboratory directors that were not only coming from very distinguished diagnostic labs, but had the vision of the future that could bring EGL into the future. I think EGL tries to be different than the typical lab by being nimble, by being able to take advantage of new research and develop rapidly new tests. I think there are two or three things that one can think of as the future. One is whole genome sequencing. The other is starting to look at genes that are not strictly Mendelian, but predisposed to more common disease. And the third is pharmacogenetics, where looking at gene variants that can cause, say, an adverse reaction to a drug. When I think about EGL being around for 45 years, I think more about the huge breakthroughs in the field. 45 years ago, we didn't understand the molecular nature of genetic disease. We didn't understand mutation basis in the detail we do now. This is before the Genome Project. It just has been massive advances in the field of human genetics. And I think EGL has taken advantage of that growth in the field to develop new diagnostic tests as knowledge goes forward.